follow-up for the material that we didn't get covered on Wednesday. And it's a fair amount of material because I wanted to talk a little bit about two more of the topics that students shared with me that we could have discussed on our Monday discussion about more examples of molecular mechanisms. And then we have our final reading assignment for the first exam, and that is to go back to really the beginning of our course. And one of our first reading assignments was to learn something about lifespan. And so we covered that topic, but now that we're talking about molecular mechanisms, it's good to go back before the first exam to dive into that in some more detail. And so I added the second half of the chapter about lifespan and aging and cancer to our reading assignment, and I'll go through that in this recording also. And so Emily picked an article to discuss, and it is another article about how phenotypic plasticity adds to the variation in a population of a species, and that could contribute to evolution. And so normally, evolution occurs by having the variation in a population of a species being based on a variety of alleles of genes. And so that's variation in the population, that we all have the insulin gene, but there's some variation in the insulin gene. We have some different alleles, and that's true for nearly all genes, that there's this variation in the population, and that is what provides opportunities for natural selection, that some individuals with certain alleles will have a reproductive advantage and a natural selection over time. And the idea here is that this phenotypic plasticity adds to the variation of the population and could lead to evolution. And the idea is, you see this variation of this phenotypic plasticity, it could be a reaction norm, a range of variation that's naturally present, allowing the genotype allowing this range of phenotypes, or polyphenism, discrete things like that, right? So it's that idea, and that's a good idea, the idea of learning a little bit about this evolution, and that's I consider it a more advanced topic. What I just stated about evolution is easily enough understood, but to really start diving into evolution and the terminology and the processes of evolution, I find to be kind of a challenging. And our course doesn't have, a course in evolution as a prerequisite, and so we're trying to get the core of it. But it's nice to have some understanding of this because you can add it to an answer. So you're explaining this topic, and in just about every example, many of the examples we talk about, a person could add, and you know what? This is valuable and relevant because it could contribute to evolutionary change. And so, it's good for you to learn a little bit of this, not because I'm going to ask a question about evolution per se, but that you can add it, to extend your answers and get a fuller answer of an example, that you provide, okay? Oops, what's going on here? So here's the article, and it's about these pupfish, and it's a certain fish that lives in one location, and it's in this desert in Death Valley, and so they seem to have recently and rapidly evolved some traits that make him more successful in surviving in really just, I think, one little pond in this desert. And so one of the characteristics is they're able to handle extreme heat. And I'll show you a list of their features. And the idea is, how could they have evolved? And there's some evidence that in ancestors or closely related species today, there is some phenotypic plasticity in some of these traits, and so perhaps there's a range of that, and then they really kind of focused on not just having that plasticity, but really evolving to have that one version of that plasticity that helped him survive better in this extreme environment. this very specialized environment. So it turns out that those varieties include several things that are regulated by thyroid hormone. And thyroid hormone is a hormone that's involved in our development and development of all vertebrates. I don't think, see much phenotypic plasticity in the thyroid hormone roles in humans, but there are examples. For example, we learned that thyroid hormone is the big metamorphic hormone in amphibians. And so in fish, the thyroid hormone plays, in some species, a phenotypic plasticity that's relevant, scientists felt, for the extreme versions in these little pupfish. Okay? And so that could lead to a kind of evolution through what's referred to as genetic accommodation. And Bryce picked an article in a previous Monday discussion that included the same concept, and so we talked about it some then. Hopefully this will sound somewhat familiar to you. And the term that was used was genetic assimilation. So both those terms are on here. It's good to have those terms. Again, I'm not going to say, hey, what's genetic accommodation? But you can bring that. You can bring some familiar to that and say a value of this phenotypic plasticity is in the species and the species' ability to survive or develop in a variety of ways, depending on the environment, and to evolve and perhaps evolved new species, for example, like that. Okay, so that's what's going on here. As these scientists are focused on phenotypic plasticity as an evolutionary opportunity. Okay, and they're looking at this thyroid hormone, and I just want to show you this because in your capstone journal, it's in our course completely legitimate to just pick this title and ask AI to summarize it for you. Not summarize it for writing to put into your capstone paper document. That needs to be your writing, but to learn what is the take-home message. In this case, what's the take-home message of this article that a student picked? What can you get out of it? And you can read the article, but it's okay in our course, to be realistic about the use of it. And so, there's a nice summary like that. So hormones, in particular this thyroid hormone, leads to a potential plasticity and in the phenotype of these fish's ancestors, and that then evolution acted on one type of plasticity that was a range of plasticity present. And it turns out that changes in the evolutionary changes in the regulation and say amounts, levels, timing of thyroid hormone is thought to contribute to that. And so then here's a summary, further, AI summary, and you can see you can dive deeper into AI mode. You can start finding the research articles that are related to this one. And I talked in class about how I have traditionally used Google Scholar to find articles that are related to an article that I'm interested in. AI is doing that some too, in a different way, trying to help summarize the article. You can ask it other things, but again, this is for your research and you're documenting that in your journal, and you have to include that you're using AI as a source for this. That's where you found it. This summary was produced by AI, right? Is this from like a review article? No, it's actually AI, just one time making a summary, a kind of review summary of that article. And so this is an acceptable tool like that, okay? So here is the text, or the text of the article that Emily picked, writing about that in more detail and talking about it. I just want to show you this, and this is a very different example, but just so you're aware of this, that certain groups of fish do undergo metamorphosis. And that means that they grow from a larval, juvenile, non-reproductive stage to a somewhat different morphology in the adult stage. And so again, this is different than the story of the pupfish, but the key thing here is, this is an example you're probably familiar with. It's the evolution of a flounder, from a fish that's swimming around with a normal shaped body, typical shaped body with an eye on each side of its head, and then it goes through metamorphosis. And I brought this up because thyroid hormone regulates this. And so it's interesting because this would be independent evolution of a role for thyroid hormone in the post-embryonic development of fish that is independent of the evolution of the post-embryonic development of amphibians and the role of thyroid hormone in that process. So we first get a role for a hormone, and then modulating that hormone leads to plasticity, and then that plasticity leads to potential evolution. And I would say that's the bottom line summary in this article. There's just a picture of a flounder with eyes on both sides of the head. So one of the eyes moves, the other side of the head. There's changes in its body in a variety of places, but that's just a dramatic one. And I just brought that up as a thyroid hormone example. So here is the types of differences. So they don't, these pupfish, I don't think have that flounder morphology, but in the same way that the flounder goes through metamorphosis, what do these pupfish ancestors go through and metamorphosis that thyroid hormone regulated that may have been fixed or featured as an adaptation of these to the little pond, hot pond they live in. And so small body, loss of certain fins, larger head and eyes like that. And a big one is this ability to survive. And low aggression, they're crowded together in this little pond like that. And this high metabolic plasticity is one of the features that is thought to be especially regulated by thyroid hormone. Thyroid hormone regulates metabolism in vertebrates. You may know of a person who has hyperthyroidism, and a lot of times they just have a tremendous appetite and a really high metabolic rate, like their body is warm, extra warm, and things like that, right? So I just brought that up, but the key thing here is, well, what are these scientists looking at for the body features of these pupfish that they looked at and thought, these are some things that could have been regulated coordinately with the thyroid hormone. Now, something to mention about this is not all these are necessarily adaptations for living in this small pond. Larger eyes and head, loss of pelvic fins. What's the advantage of that? Sometimes there are secondary consequences due to primary evolutionary change. So let's just imagine that metabolic plasticity and the ability to handle really high temperatures may have been the primary plasticity that drove this evolutionary change in these pupfish, and some of these others were kind of a consequence because thyroid hormone may play a role and in the development of these other parts. And when I saw the larger eyes and head is when I remembered about the flounders and their thyroid hormone-dependent metamorphosis. So, maybe there's an underlying relationship in some of these traits where they just go along with the real reason why, they adapted these features. So, in a really interesting article about evolution. Then Bryce picked an article, and it's similar, like Luke picked an article about the winged and wingless aphids, and that was similar to an aphid story in our book. Bryce picked an article that was similar to another story in our book in chapter 1, so this is just a good example. And it's scientists that are studying Daphnia, and they're studying a defense, a plasticity that creates a different body to have more defense against predators. So this is an example that we've seen several times, and that is that these Daphnia, and they're too small to have the traditional, I don't think they have the glucocorticoid stress response that we learned a lot about that is present. So these would be organisms that have a stress response that would probably through some different mechanisms and can be triggered by the presence of predators. In this case, it's often not necessarily the predator itself, not seeing the tiger, but seeing the evidence of the presence of predators in the environment, in this case in the water. And so a famous example of that would just be like an excretion, like in a bigger animal, like a predatory fish. They're going to produce urine. And some prey animals have mechanisms that are sensitive to the presence of evidence of the predator. But this is just a little animal. And I think the key thing from Bryce's article for this is that, first of all, that it's a little organism, simple organism responding to stress. So, really different story in the molecular mechanisms. And that's what they described here. What could be the mechanism if we're not looking at, something like glucocorticoid, corticosteroid type stress response, chronic stress response to the presence of evidence of predators? What would it be? And then the other thing is that this is can be a transgenerational effect. So the parents produce or experience this, and then sometimes can pass it on to their offspring. So a couple of things going on there, but in particular, what is the mechanism? And so here's something that is a good example, and that is this term pheromone. I think I have that written up here, maybe not in some places. I thought I did, but I think I have a slide of this term pheromone, and that is this evidence of signals in the environment that could be presence of predator. So the Daphnia is going to have plasticity that in the presence of that environment, it grows to have a different body. And in this case, the body is going to have what they call neck teeth. So it's just evidence of better defense against a predator grabbing them. Okay? And the key takeaway for me from this, I highlighted here, and that is how they wanted to find responses to this. And so what they did is they looked at all the genes being expressed, and we talked about this a little bit. Well, this is an article of talking about sort of kind of a traditional, maybe somewhat old-fashioned ecology story of, wow, look at these little Daphnia. They develop differently in different environments, presence or absence of evidence of a predator. But what they did is this very modern tool of looking at all the genes and looking at changes in gene expression to identify genes that go up and down, gene expression that goes up and down in the presence of this pheromone. Okay? And they found a bunch of genes. And if I just scroll back to the topic, the title, you see they've identified these ionotropic glutamate receptors as being involved in mediating that change in the development, where in the presence of the pheromone from a predator, the Daphnia developed with these neck teeth. So that's the key concept here, is they used a lot of different genes expression to find genes that look like they changed expression. And that's what this whole article is about. And so, some ecology about that. And then here's just what neck teeth look like. So it's just a change in their morphology, their body, that is understood to help defend against a predator. So, the things that eat these Daphnia are probably like little larval fish with little tiny mouths, and it looks like it's just harder for them to get a hold and crunch these little Daphnia if the Daphnia have these neck teeth. Okay, another picture of neck teeth. So they have those. There's a plasticity. It's a phenotypic plasticity. It's a polyphenism, yes to neck teeth or no. And they only form those in the presence of the pheromone, evidence of a predator. And the idea would be that they can do that, but why do that if there's no predator? So it's a classic example of phenotypic plasticity, where why bother to make that unless the reason is there, right? And here's just a drawing of some different examples of the of the neck teeth, that's B. But there are other stories like this in Daphnia about how the animal has phenotypic plasticity in its morphology. Morphology is the shape of the organism. And you can see, depending on environmental conditions, it can develop quite differently. Our book has an example like E, where our book in chapter 1 talks about Daphnia, it talks about the presence of a predator, and it makes a helmet like that. So there's the figure from our textbook, and it refers to that as a helmet. And you can see that looks like, to me, a more dramatic example than neck teeth, but just a totally different example of this story. And so from this we get that term caramone, and just a complementary example is Daphnia. So what we get from this is, again, the idea of, well, here's an organism that nobody's studied them, the details of the molecular mechanisms of development, but because we know so much about other species and the role of the genes in other species, scientists can look at the genome of Daphnia by looking at changing gene expression, and they can identify genes that are differently expressed in the two types, the two morphologies seen here, or plus or minus neck teeth, in this case, the article Bryce picked, and look and identify genes, and then study those genes and show which genes are important. What's the pathway, like that, okay? So then here's the last one, and this is the example that I brought up. So the example I brought up is my own research, and, it's kind of embarrassing to push your own, topic like that, right? Like, this is what I work on. But it turns out it's a very good one, and I just want to go back over some of that. So what I talked about is how that caterpillar and moth species that I study has a polyphenism, which is it can either grow up quickly during the summer months and reproduce, or it can slow down and stop development in between a caterpillar and a moth during that metamorphosis. It can stop at that stage and overwinter. And that happens in lab without any change in the temperature. So they are not overwintering because they're cold, and they're preserved like food in a freezer. It's not like that. They do the same thing in the summer temperatures. It's all about day length. And so day length is the cue. Short day lengths indicate cold is coming, and they stop in this pupal stage. So scientists have been studying this, but it almost undoubtedly requires them to extend their lifespan. They almost undoubtedly have to activate FOXO to extend their lifespan, as we saw with the story in nematodes back at the start of our course, where nematodes were the original organism that was studied in lab to understand that molecular switch to extend lifespan. So almost the same, and certainly it is, and I pause in my caterpillar species, moth species, includes that extension of lifespan. Lifespan is part of the life cycle, and so it's another example of us. This is not just embryology, it's called developmental biology, because in part we study the whole life cycle. So we're studying it here. And I put up here too, my species is called Manduca sexta. I call it Manduca. And I realized, well, I should say the common kind of general name for multiple species in this category is a hornworm. So you're welcome to call it a hornworm. That's easier to remember than Manduca. And it's a good example. And it's a good example because we're going to get an extension of lifespan, but simply we're going to have this polyphenism, yes or no. Are you going to develop fast, or are you going to stop at this point and wait for better conditions? So it fits into that category. And then we can add more to it, because I added that the reason it stops is because the animal stops making ecdysteroid. And so here we have this example where in arthropods, all arthropods ancestrally, so ancestral feature of arthropods is they molt by shedding an exoskeleton. That molting process is called ecdysis, and molting hormone is this ecdysteroid. So it's present in all arthropods, and then in insects, it got recruited to play a new role, which is regulating this metamorphosis. So post-embryonic development life cycle, it is involved in this molting, molt, like that. But now it's got a new additional role, and I studied that, and the role in that I described in molting is a big peak above a threshold of ecdysteroid. So this is a concentration of ecdysteroid with ecdysteroid concentration on the y-axis. A big peak causes molts. Each of these molts in all arthropods caused by a big peak. And then we see these smaller levels that are actually driving this metamorphic change forward. So we see that. So it's just a nice example of the evolution of this. And then we see, well, if you're driving evolution forward with this hormone, then if you just pull the plug, literally stop making the hormone, you're going to stop those changes. And so, it's a really surprising thing, because you take this traditional molting hormone that's like playing this type of role, and you get a new derived role, and then you can use that. And it goes back to, that suggestion by the authors of the Pupfish article that thyroid hormone probably played in this. So ecdysteroid plays a role in this. And so when you say stop making ecdysteroid, just so you kind of connect it to some of the other topics of the course, it's going to increase the lifespan probably. So we're going to do that, throw that switch where we're going to pause the maturation and reproductive in this organism, and we're going to extend its lifespan, because it's got to get through winter to reproduce under better conditions. So that's what's going on here. Again, hornworm is the common name, molting and ecdysis, like that. Then when you have that, another message about this is that we've seen this, and we haven't really emphasized it. When you look at species of insects, you see that this metamorphosis is evolved independently multiple times. Sometimes it appears to be taking advantage of the same role of ecdysteroid in the pupa that I study. Other times it's different stages. But in general, the thinking would be that it looks like it's pretty easy to evolve, because it evolved independently multiple times. And so there are likely points in the developmental process where, by modifying something, you could cause the organism to stop developing. And when we think about what that might be, as developmental biologists, we would say, I bet there's a hormone involved, because hormones can regulate overall things. So my story becomes a good example of how the polyphenism could evolve, plus or minus diapause, could evolve simply by regulating a hormone. I think that's what the people that wrote Emily's article were thinking about, and plenty of other examples. Then Bryce's article becomes interesting, especially because it's such a simple organism, it wouldn't have some of those same hormones. So what's the regulator there? And we'd be down more at cellular activity, so we'd see those receptors that are maybe involved in that Daphnia necti story. So putting that together like that. So, again, up here in the upper right, in this hornworm that I study, it appears that pupal diapause evolved simply by stopping synthesis of ecdysteroid. And that could lead to all these other features like that, including that central feature of, if you're going to try to extend your time before you reproduce, you better extend your lifespan. So it kind of connects to that. And then we have a really good understanding of the molecular mechanism of how ecdysone affects us cells, and it is by changing gene expression. So in Bryce's article, let's just study all the genes like that. We can study all the genes and start to get an indication of what is changing based on the presence or absence or the levels or the duration of a hormone like that. Yeah. So I did study different parts of this, and I just want to show you a little bit more of this because it shows how coordinated this development is. And so one thing I started out is I just take out their brains under sterile conditions and grow them in liquid nutrients and under sterile conditions in dishes, and that allowed me to either add the hormone or not. In the case of adding a hormone, what it's showing here is those little arrowheads point to a part of their brain that's called the optic lobe. So we're just looking at half of their brain. And the optic lobe is going to grow a lot during metamorphosis because the larva caterpillar has really simple eyes, and they're going to get destroyed by programmed cell death. And we're going to get new adult, very big compound eyes of the adult moth. And compound eyes in insects are multifaceted. They're made up of thousands of little eye units. And so this stripe here is showing proliferation in a set of stem cells. It's a kind of a horseshoe shaped stripe like this in this optic lobe, because we're going to get new input coming in here through the optic nerve of big eyes with lots of information and lots of brain processing, to have a visual image to avoid, say, predators and obstacles and stuff like that. And so, complicated vision requires more of a brain. And so really none of these dots here, these are cells in the cell cycle that are going, that are copying their DNA. So they're going through the S phase of the cell cycle, and you see that if you have the hormone and not if you don't. So just a specific way of detecting whether or not development is occurring in a dish. And I also would culture these eyes. So I could cut out the precursors to the adult eyes and grow them in a dish. And I could cut them out from this stage here. So that's the adult eye that's growing like that. And I could cut them out at different stages. And this is when metamorphosis is happening. And this is when the ecdysteroid levels drop to 0. So the adult eye is developing, and then it's going to stop. And part of the features of development of adult compound eyes in insects is that they first make a big sheet of cells, and so that's what I circled here. It's already made the full-sized eye, and proliferation is still occurring. And the proliferation still occurring is the patterning. So then there is a wave from posterior to anterior, and that wave edge is shown here. The eye is starting to get patterned. So this is just like, I use the example of, like, imagine a big plain blanket on a bed, and then you're going to quilt it into a pattern, and you start at the bottom edge and just go quilting up the pattern. So you already have the cells. You're going to make a few more cells as you make the pattern to get the different eye units, but that's going on. And this wave of development is involved proliferation, like in the brain, but several other steps as well that are really interesting to study and see in a dish, because they stop during diapause, not because it gets cold and they have to slow down. They stop because the whole story is run by ecdysteroid. So we go from, again, that molting hormone ancestral role to this more derived role in insects that go through metamorphosis, where it's driving the metamorphosis forward in an ongoing way. Take it away, it stops, add it back, it goes. And that can happen whether the animal sees short days or long days. You take this eye out from either an animal that it thinks it's summer or thinks it's fall, and it has the same responsiveness to ecdysteroid. So it just shows that. And I just show you this, that there's a connection. So here's the eye developing in that patterning beginning, and there's those stem cells. And what they're doing is they're wiring the eye to the brain on an ongoing basis. So that horseshoe shape of those stem cells is matching up this line of eye neurons that are forming and sending in a stripe, kind of a horseshoe-shaped stripe of optic nerve axons into the brain to make those connections. And those have to stop together in a coordinated way. And I could separate those and culture them separately in a dish, and I could get them to stop and go in a coordinated way because the hormone is controlling both those. So I just think it's a memorable example to see how the hormones can coordinate the development, even if the parts are not connected. And so this is just a drawing to show that. It's a drawing I made. Good job, Dave. And how the pattern is sweeping across the eye and sending in lines of neuron axons connecting to this horseshoe loop of these stem cells. Nice example. Here's just a picture of that. There's the front edge of the wave, and these are the axons being bundled by, these are each of the eye units. They're called ommatidia, each of the facets of the eye unit, and they're all bundling neurons together into optic nerves. You can see them here better if we zoom out. So that's the front edge, and you can see how they're clustered. And they are just doing lots and lots of development, and it all stops in a coordinated way, like that. Why? Because there's no hormone. So it just really underscores the big role of hormones in coordinating just all like aspects of development of a tissue like this eye and that brain, like that. It's just showing some features of that. It's a good example. And you need a hormone like this during this time to keep that going. It's all throughout this time, like that there's enough hormone to drive the development forward. And this is the stage time when the diapause happens and the animals will stop right there. Yep. All right. So that's that. And then we have our aging and cancer reading assignment. And we read the first half of our final assignment for the first exam. We read the first half back here at the start of our course because we were focused on trying to introduce aging. And then now we'll read the second half of this same reading. And the second half is more focused on cancer. So let's talk through that. And I've tried to help organize this. So one is for you to go back and look at your notes about the role of FOXO in increasing the lifespan in cells and organisms that are going to slow down their rate of aging. And orthologs of FOXO are seen in every multicellular animal and appear to be doing the same thing. So fruit flies, mice, and the original nematode that Cynthia Kenyon studied for aging and identified mutations that would affect the rate of aging. And so we talked about that this is the insulin signaling system and responding to signaling molecules that are associated with how plentiful nutrients and a healthy environment, absence of predators are, absence of crowded things, absence of harsh temperatures and things like that are all feeding into regulating this plan, like that, okay? And so just to give you a little summary of this, right, we talked about FOXO, and when FOXO gets to do its job, it's a transcription factor that increases lifespan, and it does it by this AI summary of these things, just to see, and I think I have another list of these, right? Hundreds of target genes, and some of these would be familiar to you. So, for example, it helps reducing free radicals. Antioxidants are good in our diet, so it's playing that role of increasing roles of like antioxidant to protect things. It helps protect the stability of proteins and quality of proteins. It affects the metabolic regulation to slow down metabolism. Not just to save calories, but metabolism is a harsh process. It produces free radicals. And then let's make sure we keep the DNA repaired. Let's slow down the cell cycle. The cell cycle is inherently kind of a dangerous game to play. And so FOXO genes all do many, many different things that we know from a variety of different systems. Yep, I bet that would help the health of an organism. And we're slowing things down because we're essentially, the primary thought is we're not going to reproduce. It's not good conditions for reproducing, so let's live longer, okay? So a little summary of that, try to dive in your ability to summarize the molecular pathway for everything we've talked about. So, go back, and now that we've had these examples from students and myself, about specific things that are happening about features inside the bodies of animals and plants to regulate phenotypic plasticity. Learn more about this as you learn more for our test. So that's a reason why we're going back to this. I intentionally did this thing to try to introduce the topic of aging early in our course with some reading and then dive deeper into molecular stories about it. Okay, so in the chapter, both that first and second half of the reading, because I'm kind of combining those here today, our authors emphasize some things. And so this is a good summary I put together about what does the author want to talk about that aging can involve, right? So we just talked about ways to slow down aging, and here's some features that our author brings up. And one of them is that wasn't mentioned, I don't think, in that FOXO summary, is that aging can involve changes in DNA methylation. And so we talked some about DNA methylation and how DNA methylation can decrease gene expression. So that can play a role in phenotypic plasticity, and it can play a role in aging. So the first example is one we've talked about that we've used, and it includes like the story of the queen ant, right? So as the animal lives, this is an example of what that isn't necessarily a story of aging, but just as it lives, natural changes in the environment can affect DNA methylation. So we have that as a good example. That happens normally as the animal ages, is there changes in DNA methylation. And what we want to do here is hang on to our examples we already have of how DNA methylation can contribute to phenotypic plasticity. The queen ant and the worker ant have different amounts of DNA methylation, and that occurred during their aging, right, as they grow up. And that's normal changes due to environmental factors. And so the author has some examples of that, and then throughout the book. And then here in this part of the book wants to say that aging or that living also includes just an overall increase in DNA methylation. And that increase in DNA methylation can lead to a decrease in gene expression, and therefore a decrease in the functionality of tissues. So aging can include, your tissues just aren't working as well. And that can be due in part to DNA methylation. Then also as we age or live, go through the life cycle, there can be, in addition to increases in DNA methylation, there can be errors. So I think this is just a summary of these DNA methylation that we want to get out of this, hanging on to our original examples and then diving into aging and an increase and error, and also separately errors in DNA methylation. So the author shows this. This goes back to chapter 2, and this is just that normal story. We have several normal stories like the ants, and this is like a change in behavioral due to the conditions that the female, the mother of offspring, is behaving toward her pups, her babies, like that, right? So this is an example of normal, just to highlight that. And then here in chapter 8, the author is talking about aging twins. So these are identical twins. I think they call them monozygotic twins here. And they say, these twins, identical twins, when they're young, they have really similar patterns of DNA methylation, and when they get older, they start to have some differences. And so this is just showing a pattern of the position of DNA methylation in whole chromosomes, just to give you that global feeling that there are changes over time. Now, what we would say is that the author is talking about some normal DNA methylation, right? And then this is some additional changes, right? And it can be changes due to this overall increase that then leads to differences because there can be errors. So I think the author is saying, we see this methylation, and the author doesn't do a good job explaining, this includes that normal differences, because these twins could have different environments and have differences in DNA methylation as an adult. That happens. But then in addition, there is this overall increase that could lead to aging, and that could be different in the different twins, and then errors like that. Okay, so then here is an example of age. Then so this is hardening of the arteries, and how DNA methylation increases under not just overall, but under certain aging conditions, and it goes hand in hand. What's the cause and effect relationship here? And the author's just pointing out the fact that it occurs. The errors in DNA methylation, the author uses this phrase, this term, random epigenetic drift. And the idea is that when DNA gets copied, we have to copy the DNA and the AGCT. And there's a lot of proofreading mechanisms to make the AGCT letters very accurate. There's also DNA repair. And it turns out that DNA methylation also has to get copied when a cell divides, goes through M phase and duplicates its chromosomes in S phase. The DNA methylation has to get copied also. And the DNA methylation does not have good proofreading or adjustment, repair. And so as a result, we see random epigenetic drift errors. And there are random errors that are being made. And it's a lot more of this than there is of DNA mutations. And that author is emphasizing that plays a role in aging. And so, although it wasn't summarized in Foxo's story like that, directly, I think the Foxo story pointed out, there's going to be a decrease in the cell cycle. And if we don't decrease the cell cycle, we're going to be cranking through cell cycles. And normal cells just have a mortality to them, separate from, the organism. If you take the cells out, they're not going to live forever. And part of it is, as they divide, they start to just lose the health because of that. And so here it's showing just a specific example in our book of an increase in DNA methylation. And the author wanted to point out, there's an overall increase in DNA methylation that's going to lead to a decrease in the function itself. But then there can be a component of this that's random, errors in DNA methylation leads to not just an overall decrease, but random variation in DNA increase. And that leads to aging and the different rates of aging randomly. So overall DNA methylation, and it's used to, could be used as a test to estimate someone's age, like that. And so just a little summary statement there. Errors in DNA methylation can lead to some cancers that don't have, this is moving on. So errors in DNA methylation. So that's a specific, we let up here. Then here's another thing the author brings up, because the author starts to write about cancer in the second half of the chapter that we're focusing on now. And that is that sometimes in certain very specific cancers, especially this type, and you could learn either one of those terms, there's a type of teratoma that is in the gonads, in the developing testes or ovaries, that is a solid tumor cancer. And it turns out that a person can, through some manipulations, can cause this cancer to go away, because it's not caused by DNA mutations. It's caused by errors in DNA methylation, causing some genes to be repressed and leading to this cancer. And those methylation patterns can be corrected. And so the author wants to use that here as an example of this, and they call it a teratocarcinoma. That's another name, all the same, so just learn one of those names if you want to use this example. that aging can lead to errors in DNA methylation that can actually lead to certain kinds of cancers. And there's evidence that changing DNA methylation plays a role in lots of cancers. This is just a striking example because it's the cause, because you can fix it and go back to normal healthy cells in this particular example of cancer by just correcting the errors in DNA methylation. Okay? Then see, here is another example. It's different, and this is the example that a variety of cancers have changing patterns of DNA methylation. So that can also, that error, that failure to have good proofreading can also contribute to cancer. So cancer is an extreme example of tissues going out defective. And cancer is considered to be largely a story of aging. There's a lot more cancer in older folks than younger folks. And so those cancers associated with old age are the focus of the author here is diving into some of the mechanisms of aging to understand more about, what does FOXO do? And there are other players. The authors bring up TOR. We talked a little bit about TOR, and the authors talk about TOR in this chapter. TOR has a kind of has an opposite role of FOXO. TOR is going to lead to increase in ribosome activity, increase in metabolism, increase in moving toward reproducing and not living long, like that. Yep. So, that's this DNA methylation. So we pick up several stories of DNA methylation, and we want to be careful not to confuse those with the ones we already learned. Okay, so I tried to summarize that, and what the author is trying to emphasize in this chapter, that is to not confuse you, right? We're far into our course enough now that we can keep those separate stories of DNA methylation in queen ants and worker ants and so forth separate from these examples that are put in this aging and then that are included in this cancer as a part of aging, like that. So in this, I just want to help navigate through this new reading. There's those pieces of DNA methylation. And lots of people consider DNA methylation an example of something called chromatin remodeling. Chromatin is just the DNA with the proteins bound to it. And because it's a modification of DNA, some people include DNA methylation as an example of this chromatin remodeling. And this chromatin remodeling leads to increases or decreases in gene expression. And so aging can involve changing in DNA methylation that we just talked about. And then the author just has one section on one slide that it can also include other stories in this chromatin remodeling. In general, chromatin remodeling gets replicated when the DNA gets replicated. And again, it doesn't have great proofreading. There could be errors that build up over time. And so here is the same kind of thing, looking at a different type of chromatin story instead of DNA methylation. Oh, here's the first one. This is DNA methylation. So that's just that data that we're already familiar with, that it's going to increase over time and twins are going to become different, partly due to differences in their natural environment. They're showing some phenotypic plasticity here, but also due to random errors. They're just going to start showing up with differences. And then these are just, and we don't dive into these in detail, these are just different examples. And what the author is doing is saying, yeah, there's some validity to this focus on the error rate in DNA methylation and chromatin remodeling that occurs over time, right? We have to start getting these older people have some differences, some errors that would lead to poorer quality tissues, like that. So DNA methylation changes in other stories, and both those can be normal changes due to environmental factors. So we've talked about DNA methylation. We just haven't talked about this with regard to normal phenotypic plasticity occurs. So yeah, there's going to be changes over time, over the lifespan, you bet. But author wants to emphasize here that these are errors that can lead to that, okay? And then the next thing is the authors want to bring in cancer as a age-related disease. And again, the lifespan increase is going to decrease age-related diseases. So that becomes relevant in this story of lifespan. The author really likes the idea of studying an increase in lifespan to just study all kinds of things like heart disease and diabetes and cancer, as those would all decrease if we could just increase our lifespan. And cancer is a big story in development gone wrong. And so, the author's background is in developmental biology, so tying this into our course makes sense. For us, we're starting to move toward the second half of our course, which would be this disruption in normal development, because cancers are often, they're not normal development, there's some kind of disruption that's occurring. So there could be an environmental factor, a carcinogen, causing the cancer. And so we'll get into that in the second half of our course. And so, this reading is also in part transitioning it to our second half of our course. I like that. So this just shows that there are certain diseases that have what are said to be a high concordance rate. And this is in monozygotic identical twins. And it's showing that there are some, over their lifespan, there are some phenotypes that show a high concordance. So, for example, autism and celiac disease show a high concordance. And what that means is if one twin has autism, the second identical twin is very likely to have autism. So it seems to have a very big genetic component. And then low concordance would be these probably caused more by environmental factors rather than genetic factors. And you can see out here that there are some diseases that have pretty good concordance in late life. And so the author is showing that in aging, parts of aging show a strong concordance, a strong relationship with the genetic similarity, genetic basis. And we're looking at twins as an example to understand the larger population and the role of genetics versus environment in these characteristics. And if we just go back to here, there's an increase in age-related diseases, including cancer and other diseases. And some of those, the author wants to emphasize, some of those could be have a genetic component, and some could just be due to the different environment. Were you a cigarette smoker? Well, you're going to have a higher incidence of lung cancer, as an example. Okay? So the author just has parts of that, and that's all I want to add for that is really diving into some cancer biology. And then the author really wants to go into, because they're developmental biologists, and this fits with some traditional developmental biology, they really want to take what is the traditional view of cancer, which is correct, and has taught us a lot about cancer biology, and we were just using here, that there are mutations that can lead to cancer. So there are mutations that lead to activation of oncogenes, and that's definitely the case. And this is kind of an unusual phrase, but it's somatic mutation hypothesis, and we could just focus on they're caused by mutations, like that. And the author wants to bring up a role for the tissue environment. And so what we want to talk about here, and we haven't talked about in our course, is, I mentioned it once, but here's a cell, and it's in a multicellular organism, like that, but it's still an individual cell, the basic unit of life. And so when we talk about the environment, the author wants to emphasize, well, to us, we think of the environment as what the author calls the external environment, but the cell is really responding to the external environment and the internal environment as being the environment. And so the author wants to start introducing that mindset in this chapter and uses this tissue organization field theory. And what that's emphasizing is that this cell could be cancerous because of what's happening in the cell, mutations, and also the surrounding cells can contribute. And so that's an example of what the author wants to talk about with regard to this course, Eco-Devo. And the author, is kind of an extremist, saying, well, yeah, Eco-Devo could be external environment, it can also be the internal environment. And so we got to dive into that a little bit here, and that's what's going on here, is that cancer biology can also be a story that includes this surrounding cells, like that, okay? And so this is just going to be the author emphasizing that we'll have two cell types nearby, and the author will use this, the epithelium covering skin, but it also covers like organ, like the stomach. So the stomach organ has stromal cells and epithelial cells as general terms like that, and is just describing experiments to set up this understanding that cancer could be due to the mutations, or it can be contributed by the surrounding cells. There can be a contribution of the surrounding cells. And there's good examples of that, and it must involve some kind of communication between the cells. And, we've talked about that in our course, that a lot of these pathways involve cell communication, like that, okay? So here's just a summary of that, right? AI summary. Like, we got to understand this a little bit. So tissue organization field theory taught, really emphasized by a couple of leaders in this, and here's just a summary of it. And in this case, I wouldn't say, a summary to help you produce, memorize the number, the list, but just help you to understand what are the features that these people are talking about, to be able to describe it in words on our exam, to distinguish this idea of cancer involving communication between cells, right? And so our author is enthusiastic about it and dives into it a little bit too much, I think, because, again, this is all about, the internal environment includes just surrounding cells, because ultimately it's a cell that is the basic unit of life. And it's a really great point to make. It just becomes kind of a confusing story to make. But cancer biology is a big story, and it's part of our aging story. And so our author wants to emphasize that the tissue environment is going to age also, and can lead to age-related diseases. And the first one is cancer, okay, like that. And this is just author bringing in another kind of tissue story, and that is, here's a tumor, and it needs blood supply. So in this case, just a different story coming in with regard to the development of cancer. And so then the last thing, so that issues between neighboring cell populations. So, what we get from that is, it's not just an external environment, it is an internal environment story too, because cells are the basic unit of life. And that can contribute to cancer, like that. And then the final thing we want to get out of this is another tissue level thing, and it's the aging and this senescent cell phenotype. And the senescent cell phenotype term the author uses is chronic sterile inflammation. So inflammation that's sterile inflammation would be like when you twist your ankle or knee and you get swelling. That's inflammation, but it's sterile. There's no infection, right? That wasn't caused by bacteria, but it's inflammation, and the immune system is involved in regulating that, and it is temporary. Your body heals, the swelling goes down. So this is chronic sterile inflammation, and it's associated with aging. So another example of aging, the author wanted to bring up, that must be modulated when we regulate the rate of aging and lifespan. And so that's our final example in this list of features that are covered in this chapter. Just to briefly describe it, the author describes that this includes that there are some cells, that the individual cells should go through apoptosis, programmed cell death, and be cleared out. And we should keep removing these unhealthy cells from this tissue, so a group of cells, because they are decreasing the health of the tissue as part of the aging process. So some older person with some creaky joints, but just all different kinds of parts of their body could have this chronic sterile inflammation. Okay, and so big pathway like this leading to chronic sterile inflammation like that. Here's the youthful pathway, right? So issues come up and we get them taken care of, and we have a temporary sterile inflammation. And if we don't get those fixed, we'll have it chronic, and it will lead to these. So these contribute. And so that's our last piece from this chapter. Here the author is putting those together in an image, and that could contribute to cancer, to this chronic sterile inflammation situation. It can also contribute to cancer biology. And so it ends up getting kind of complicated. I want us to read through it to add molecular mechanisms to our stories that we can describe with regard to lifespan, because that was our first story in our course, and it's a really good story that's really continues to drive this field forward. Eco-devo in lots of field of biology are really driven forward by biomedical research in human health, like that. It's a big motivator for trying to understand these principles. And so that's where we're at. We'll have a review, so please bring questions on Monday to class about trying to understand all this stuff, and we'll go from there for our exam next week. Okay, thanks.